All right, folks, the clock on the wall is starting to trick me. I can't rely upon it as much as I should the computer. The computer says I'm a minute late. I'm sorry. But let's start talking about the central dogma of molecular biology. Boy, this is going to be fun. Thursday, even more fun. But boy, today's going to be fun. So here's where we are, right? We've talked about Gregor Mendel's work, and then Thomas Hunt Morgan comes along, and there's particles that are inherited. There's information that gets passed from one generation to the next. Those particles, in a sense, reside on chromosomes. And now we're getting to the point where we can start to talk about how not just those genes are arranged along a chromosome, but how that information actually gets tapped into and used. So today, the title for this lecture, and it's going to be the same title again on Thursday, the title for the lecture is The Central Dogma. It's the central dogma of molecular biology. And in a nutshell, what the central dogma is all about is how it is that information gets taken from where it's stored in DNA and ultimately then used to make proteins. Now, the only other place you're going to find a dogma is in a philosophy or a theology kind of class, right? Molecular biology is kind of unusual, and the history behind the word dogma in this context is also kind of humorous. You saw, if you came in a little bit earlier today, a video of James Crick, who was talking about his role with the Nobel Prize winning work to get the structure of that double helix worked out. If you watched the video, if you picked up on it a little bit, he's got a sort of quirky, weird sort of sense of humor. And there are times that I think when you hear him talk, he's not that smart. He was just in the right place at the right time. Maybe he had some stuff that clicked. One thing that unequivocally he made a mistake on, and again, this is the guy you heard talking who got a Nobel Prize. One thing he made a mistake on is he went to a dinner at Harvard shortly after having gotten the Nobel Prize, and a bunch of graduate students were sitting around, soon-to-be PhD types from Harvard, were sitting around at this dinner drinking wine and cognac and brandy and smoking cigars, and they get him lubed up. He's loose, feeling pretty good, and they see him. So, Dr. Crick, Laureate Crick, tell us what other things you're thinking of that you think are going to be really important. What's your next big insight in terms of how the world works? It's sort of the Super Bowl equivalent of, you've just won the Nobel Prize for figuring out the structure of DNA. What are you going to do next? And what he says is I got this sort of idea, I'll call it a dogma, that there's this central dogma that information in cells flows from DNA to RNA to protein. And the people in the room, just their jaws drop, is like, oh my gosh, Nobel laureate Crick just said that there's a dogma that information flows from DNA to RNA to protein, and these guys know what the word dogma means, whereas Crick doesn't, apparently. They know that dogma means it's literally been handed to you from God. Right? This is, a dogma is information. It's a rule. It's like Moses' commandments. This is something that's come directly from God. And Crick is telling us that God has told him that information flows from DNA to RNA to protein. And so they leave there, and they hit, there's news stories that come out about this. It's supposed to be a quiet, casual conversation. But this is such big news that the rest of the world in a couple weeks knows about the central dogma of molecular biology. And Crick is saying, like, well, why is this such a big deal? I don't get it. I said it was a dogma, and somebody explained to him, dogma, so how did it happen? Did you have a dream, or was there a vision when God was telling this to you? It's like, what are you talking about? I said it was a dogma. That means just a wild guess. I don't really have any idea. I was just thinking that might be the next thing. No. Dogma means that God told you. He's like, really? It's like, oh, my bad, right? So He thought he was just making a guess about this. He called it a dogma by mistake. The name stuck, and we now, as molecular biologists, have the central dogma of molecular biology, because despite the fact that when he first talked about it, it was just a wild guess, a shot in the dark, it turns out it is, in fact, central to everything that molecular biologists do. It's a good orienting, sort of guiding principle. This idea that information is stored in DNA, it flows through an intermediate molecule, RNA, and then ultimately that information gets used to make proteins. And it's proteins then that do the stuff that living things need to do. Catalyze reactions, build structures, things like that. That whole concept is the central dogma of molecular biology. And again, if these Watson and Crick guys were perhaps a little bit better read, they would have called it something else, but we have now something that effectively is dogmatic. It is central to the whole thinking of molecular biology, and that's what I'm going to talk with you about today. So, how do we get there? We got Mendel talking about particulate inheritance. So you got Morgan linking particulate inheritance to chromosomes. You ultimately, and we talked about this in class on Thursday, you ultimately have Watson and Crick coming along and saying, here is the structure of the molecule. And once again, structure and function really are interrelated in a biological setting. You've got the structure of DNA. How is it now that with this structure, knowing that particulate inheritance is residing on this molecule, how does this molecule store information? That's the big question for where we are right now. The first insights, again, come from Mendel and Thomas Hunt Morgan. Then moving the ball a little bit further down the field was another British physician, Archibald Garrod, in 1909. He's a doctor, he's a physician. He's got this family that comes to him, and they say, you know, we've got this embarrassing problem. It turns out that every time that we urinate in public, people laugh at us. All right? And the physician, carrying Archibald Garrod, he's worried about this. Why are people laughing at you when you urinate in public? And they say, well, it turns out nobody else's urine changes color. Ours becomes black shortly after we've urinated. And Archibald Garrod says, okay, I see why they're laughing. That's kind of weird. That's very strange, isn't it? And the people in the family said, we thought that's just what everybody's urine did. It turns out we're the only people that we know who have black urine. And so Archibald Garrod studies this a little bit. And what he ultimately discovers is they've got a condition known as alkaptonuria. It wasn't unique. There are other people who had the condition. It just apparently hadn't talked about it as much. But he found that it ran in families. And he found, based on what he knew from Mendelian genetics, he found that families that had it, individuals who had this trait, whose urine would turn black after it had been exposed to oxygen, they were homozygous for a recessive gene, right? Knowing something about Mendelian genetics, looking at these families, studying their pedigrees, who had the trait, who didn't have the trait, who was likely to have it when they had children, he found that this particular trait, alkaptonuria, urine turning black, was due to an error in metabolism, further that it was something that was heritable. And ultimately, he was able to show that the error in metabolism was they were not able to make one particular protein that everybody else who didn't have alkaptonuria could make. All right? And so Archibald Garrod, with those sort of three strands floating around, this is a heritable trait. The people who have it can't make, they have an error in metabolism. And the third is that this is due to a defect in a particular protein. He ties those three strands together and he coins a phrase that we still use often today. It's this idea of one gene, one polypeptide. Did that make it to the outline? It did. Look at that. It's like I prepared this lecture in advance. How about that? So Archibald Garrod gives us this idea, one gene, one polypeptide. The backstory is kind of interesting. He's dealing with this family with this embarrassing urination problem that they've got. Maybe that'll help you remember how this comes to be. But he says that, for every gene, there's a protein. He's the guy who's making the connection that genes and proteins are related, and we get to this idea, for every gene, there's a polypeptide. Now, I'll tell you today, that is an oversimplification. And as we go through today's lecture, but especially on Thursday's lecture, you'll see how there are some instances where a gene might actually give rise to multiple proteins, right? And you'll see some instances where genes do something important without giving rise to a protein. But as a nice sort of simplification to start with, and it's good for 98% of all cases, for every gene we've got, you make a protein. Human beings, you and I, we've got on the order of 32,000 genes, okay? And you know what the sum total of all of our genetic instructions is called? We call that a genome, right? So the human genome has 32,000 some genes within it. The Archibald-Garrod sort of extension here, one gene, one polypeptide, works out to something like this. If you've got a defect in any one of those 32,000 genes, you've got an inborn error of metabolism, and that will manifest itself in some way that makes you different from everybody else. Maybe your urine changes black when it's exposed to oxygen. Maybe you're a diabetic, right? There are a whole bunch of different things, but a mutation in one of those 32,000 genes probably results in an inability to do something that people who don't have that mutation can do, okay? That's the whole idea of one gene, one polypeptide. And it might also cause you to think that there are maybe on the order of 32,000 different genetic disorders, right? That's a reasonable extension of that thinking. I don't know that you'd be that far off the mark if you were thinking along those lines, right? What makes you a little bit off the mark is the fact that some of those genes are so fundamentally important that if you have a messed up version of it, we don't see you sitting in classrooms because you're dead, right? So you can take maybe 10,000 of the 32,000 off the list because a problem with one of them is just incompatible with life. You can add more to the list because there are multiple ways for genes to be messed up, and each messed up version could give rise to a different kind of genetic disorder. But more or less, one gene, one polypeptide, one mutation in a gene, one genetic disease. It's a reasonable way to think your way through these things. So, Archibald-Garrod gives us, this is his gift to the world, this concept of one gene, one polypeptide. And the next people to sort of pick up that ball and try to carry it a little bit further are a pair of Stanford researchers named Beadle and Tatum in the 1940s, during World War II. They played around with a bread mold, Neurospora crassa. So a bread mold, the sort of stuff that grows on your Wonder Bread after 3-4 weeks sitting on your kitchen counter, right? So literally a bread mold. The particular bread mold that they're studying was of interest for a couple of reasons. One of them is that it didn't need a lot to be able to grow. It needed a carbon source, it needed a nitrogen source, it needed water, and it needed some sort of energy source, and that usually came part and parcel with the carbon source. Sugar is what it was coming down to. But if you fed Neurospora crassa, this bread mold, those 4 sort of fundamental, very basic things, it could do all the rest. It could make all the, I didn't say anything in that list, did I, about an amino acid? Glycine, valine, alanine. I didn't mention any amino acids. It can make all of its own. How about that? That's a pretty cool trick. So this is an interesting organism. It can make everything that it needs from those 4 very simple things: water, nitrogen, carbon, and an energy source, right? It can do everything else. It can make every other molecule it needs from those very basic building blocks. And so Beadle and Tatum are studying Neurospora crassa for that reason, and they find, after a while, that they find there are some Neurospora crassa bread molds that can't make everything, that instead need to have their food supplemented with something beyond just those four very basic things. Like maybe they need a particular amino acid. If you don't also feed it the amino acid, these bread molds can't continue to grow. And they found that these organisms that couldn't make a particular amino acid, for instance, had that as a heritable thing. It wasn't just this bread mold, it was this bread mold, and all the baby bread molds that came from that bread mold would continue to have that problem. In the vein of Archibald Garrod, one gene, one polypeptide, Beadle and Tatum say, hey, these guys have an inborn error of metabolism. They have a defect in a particular protein that prevents them from doing a particular aspect of metabolism. It's heritable, this defect. That protein must be spelled out on some part of a chromosome, and that's being passed on from one generation to another. So again, this whole idea of one gene, one polypeptide. So bread mold normally could grow on what they called minimal media: water, nitrogen, carbon, and energy, right? But they found that they could get some mutants that needed something more, right? Minimum medium wasn't enough. And one of the first ones that they really started to study were bread molds that needed to have the amino acid arginine included in their food. All right, so these bread molds can do everything else you need. They can make valine, they can make lysine, they can make purines, they can make pyrimidines, they can make lipids. They're cool for everything, but they can't live unless you also feed them the amino acid arginine. Right? So they find this interesting mutant. It needs the amino acid arginine, is part of its food. If you give it arginine, it's just fine. And they beat this guy up. They beat this bread mold up. They find similar mutants on a number of occasions. And what they find is that there are a couple of different ways that these bread molds can be defective in their metabolism. All of them can live if you give them arginine. But some of them can do fine without arginine, but only so long as you feed them citrulline, a molecule that chemically looks very similar to arginine, but is different from arginine. Others can do just fine if you feed them ornithine, or citrulline, or arginine. But some of them, if you feed them ornithine, can't do anything by themselves. Or better, if you feed them ornithine, they still have a problem. What they find out from characterizing these different kinds of arginine-dependent bread molds is what they're finding is they're figuring out a metabolic pathway. How is it that the bread mold usually makes the amino acid arginine? It turns out that the process involves first making this chemical ornithine, and then converting the ornithine to citrulline, and then converting the citrulline to arginine. All right? So it's a three-step process. Each of those processes requires a specific enzyme that catalyzes that reaction. Okay? And what they're finding is different mutants. One that's doesn't have the enzyme that allows it to convert citrulline to arginine, another that doesn't allow it to convert ornithine to citrulline, and another still that doesn't allow it to be able to synthesize ornithine in the first place. All right? So, once more, one gene, one polypeptide. They come to this idea that there's at least 3 proteins involved in the metabolic process of creating arginine. And if you have a defect in any one of those, you can't make arginine. It's a stepwise process. And these are heritable. So once again, one gene, one polypeptide, inborn error of metabolism, and they get a Nobel Prize for figuring that out, but also figuring out the metabolic process by which arginine gets made from scratch. Okay? So here's another way of drawing out what it is that previous slide showed you. Those were experimental details. Here we're simply talking about the fact that there are 3 genes involved, the gene A, B, and C. If you have a mutation in one, you can't do one step in the process. At the end of the day, if you feed them arginine, they're all okay. All right, so Watson and Crick come along with this structure. They're living in a world where people are talking about one gene, one polypeptide. And then this bomb gets dropped about the idea of the central dogma. On the screen now is, in a nutshell, what's up with the central dogma. Once again, information is stored in DNA. It flows from DNA to RNA, and then the information in the RNA gets used to make proteins. This is an awfully simple looking slide, isn't it? There's a lot of stuff under the hood with this particular slide, a lot of fun stuff that I just can't wait to tell you about, and I'm tempted to just spill the whole beans to you right now. But let's take it a little bit slowly, right? There are arrows here. These arrows are shorthand for some pretty complicated, pretty important processes. The arrows have names. This first arrow, taking the information from DNA and using it to make RNA, that process, this arrow, in a word, is called transcription. Okay, so transcription is the process by which, in vocabulary, right? Transcription is the process by which we take information that's stored in DNA and use it to make RNA. The next arrow also has a name, this process. Okay, it sounds similar to the name for the first one, but it's different. Don't confuse the two. It's like fingernails on a chalkboard to a molecular biologist in particular, but probably biologists in general if you confuse these two. The second arrow is translation. That's the process by which you take the information stored within RNA molecules and you use it to make proteins. So transcription is the first arrow, translation is the second arrow. Don't confuse the two, please. If you confuse the two, don't tell anybody that you knew me and I had anything at all to do with this. It's embarrassing. I don't want to be a part of your confusing those two words. So transcription and translation. They're catalyzed by, guess what? How do you do transcription? What's the cliche answer? Oh, a protein does it, okay? Well, we've got a name for that protein. The protein that catalyzes transcription, excuse me, is an RNA polymerase. Show you pretty pictures of RNA polymerases in just a little bit. But that's the name of the enzyme that catalyzes the reaction. How do you suppose you do translation? There's a cliche answer for that. A protein does it, right? What's the name of that protein? That protein is called a ribosome. So ribosomes catalyze translation. RNA polymerases catalyze transcription. Very different chemical processes, very different enzymes, sort of similar sounding names, transcription and translation. All right, so there's more of the details of what's going on here. Let's talk now about how it is that you actually might be storing some information in DNA. And that gets me to talking with you about the genetic code. Before we do that, let's take just a look in terms of the context of what's going on here. So these two pictures are showing you how it is that transcription and translation play out inside of two different kinds of cells. The cell on the left is a prokaryotic cell. The cell on the right is a eukaryotic cell. Okay? You guys remember the big difference between prokaryotes and eukaryotes? Prokaryotes are simple, eukaryotes are complicated. Prokaryotes have no internal membrane-bound compartments. Eukaryotes have internal membrane-bound compartments. Among those internal membrane-bound compartments is the nucleus. Within prokaryotes, transcription and translation happen at the same place at the same time. Okay? Come to grips with that. The enzyme that's transcribing DNA to make an RNA copy, RNA polymerase, it's doing its thing at the same time that ribosomes are sniffing around looking for RNA molecules to translate into proteins. As RNA molecules are coming out the butt of an RNA polymerase, ribosomes are right there waiting to really take a close look at that RNA to take that information and use it to make a protein. Transcription and translation in prokaryotes are happening at the same place at the same time. One can't take place in the absence of the other, also going on pretty much at the exact same time. So that's the story for prokaryotes. But again, eukaryotes have one other thing that's going on. We've got this funky nuclear envelope, the double phospholipid bilayer packaging, the bag within a bag within a bag that is our nucleus. And one of the things that eukaryotes do as a result of having that nuclear envelope is we separate in time and space transcription and translation. Transcription happens exclusively in the nucleus within eukaryotes, and translation happens exclusively in the cytoplasm, outside of the nucleus. So whereas in prokaryotes, transcription and translation are happening at the same time in the same place, eukaryotes do something a little bit different. We segregate them. Transcription happens in the nucleus. Translation happens in the cytoplasm. Turns out that's a huge deal. It's a really big deal. The main reason that it's a big deal is this. Eukaryotes can change the information content in their RNA molecules before ribosomes see them and start to do translation. In prokaryotes, that can't happen. Whatever's spelled out at the level of DNA, that's what ribosomes are ultimately going to see. But in eukaryotes, because transcription happens in one place and translation happens somewhere else, there's at least an opportunity for cells to change the information content in their RNA molecules relative to what was at the DNA level before ribosomes start to do translation. Right now, that may not sound like that big a deal. It ends up being huge. I'll tip my hand a little bit, and I'll tell you, here's one of the reasons why it's huge, right? I told you a little bit ago we've got 32,000 genes, right? You might think we can make 32,000 proteins. Guess what? You can make on the order of 250,000 proteins, 250,000 some proteins. You know why? It's because for us, because of this information processing that happens in our nucleus, we can take a single gene and use it to make multiple versions of proteins. All right? We'll talk a lot more about that on Thursday. But how about that in terms of an implication? For every gene inside of a prokaryotic cell, you make one protein. For every gene inside of a eukaryotic cell, you can make at least one protein. But because of this RNA processing that can take place in the nucleus, we can actually make much more than one. All right, so there's the lay of the land. Again, how is it that Watson and Crick's structure, this double helix, lets you actually store information? What's the nuts and bolts of the information being stored? Let's talk about the genetic code. So once again, we've got here another way of taking a look at the central dogma. Okay? On the top of the screen, you've got blue double-stranded DNA molecules. In the middle, you've got a red RNA molecule. And at the bottom, I'm going to call that purple, you've got a purple string of amino acids, a polypeptide, if you like, a protein. Information flows from DNA, stored here in a DNA molecule, through transcription to an RNA molecule, and the information in the RNA molecule with translation gets used to make a protein. Well, we've talked about this a couple times already. How's the information stored in the DNA molecule? Let's take a little bit of a closer look one more time at the DNA molecule. Remember, I talked with you about this on Thursday. We've got double-stranded molecule. The information on one strand is redundant with the information on the other strand. Cool. For every A on one strand, there's a T on the other. For every G on one strand, there's a C on the other. Notice how they're helping you out here. They're telling you G's are bigger than C's. This is a purine. This is a pyrimidine. Fills the space nicely. Look at that. A's are purines. T's are pyrimidines. Fills the space nicely. It's uniform all the way across. But for every A, there's a T. For every G, there's a C. So we've got two strands. The information's redundant. The strands, again, I've mentioned this on Thursday, they are anti-parallel. So they're not just parallel, one line here, one line here. They're anti-parallel. Their orientation is opposite of each other. One of them runs 5' to 3' from left to right. The other runs 5' to 3' from right to left. Okay? You know what that 5' means? The 5' means that at the level of the ribose sugar, remember nucleotides have three parts. They've got a nitrogenous base, they've got a ribose sugar, a 5-carbon sugar, and they've got a phosphate group. The nitrogenous base gets attached to the ribose sugar at the 1' carbon. So going clockwise around that ribose sugar. The first carbon that you see is what we call carbon 1. Thank you, organic chemists, for an imaginative name. Right? So the first one you see, 1'. Next one around, 2'. Next one around, 3'. 4'. Last one around, 5'. 5-carbon sugar, right? 1, 2, 3, 4, 5. We got all 5 accounted for. The 3' carbon has a hydroxyl group dangling off of it, an OH. The 5' carbon is the one that the phosphate group gets attached to. 1' gets the nitrogenous base, 3' has a hydroxyl group, 5' has a phosphate group. At one end of a DNA molecule, it's the beginning of the DNA molecule, the strand will have a ribose sugar that has a phosphate group on it. And it's not connected to anything else because it's the first one in the chain. The next one in the chain gets attached to the first one by a phosphodiester bond between the phosphate group of the second nucleotide and the 3' carbon of the first nucleotide. Okay, so the first nucleotide that you link into this chain to make this polymer that we're going to call a DNA strand has nothing on its 5' carbon but a phosphate group that's flapping in the breeze. The next one, the next nucleotide, its phosphate group is tied up in a covalent bond with the 3' carbon of the first one, and its 3' carbon has a hydroxyl group that's flapping in the breeze with nothing to do until the next nucleotide comes in. And you keep adding nucleotides on to the 3' end, making a new 3' end all the way, keeping that 5' end fresh. That phosphate group is just hanging out there. So that's this orientation business. You got a 5' end, 5' to 3', and the other side, you got 5' to 3' running in the opposite direction. Here the phosphate group is dangling out there. Here the phosphate group is dangling out here. Here you've got a 3' hydroxyl. Here you've got a 3' hydroxyl. That orientation is, for I'm sorry to say, I appreciate it's potentially a little bit confusing. It might help you to look at a picture of a nucleotide again, maybe zoom in on the 5-carbon sugar, the ribose, to make sure you see what I'm talking about here as I'm waving my hands to you this morning. But I'm sorry to say that that orientation, 5' and 3', turns out to be a really big deal. It's a big deal to molecular biologists. It's a big deal to cells. I distinctly remember the first time that I heard somebody telling me about 5' and 3' and how it was a big deal and it was something that I should remember. And I remember thinking to myself, I hope they're wrong about that because I don't want to wrestle with it. Turns out I needed to wrestle with it the next time that I heard about 5' and 3' because I didn't do well on the test that came up after the first time I heard about 5' and 3'. So it's worth taking a little time to make sure you're comfortable with the difference between the 5' end and the 3' end of a DNA molecule. It's big. It's a big deal. If nothing else, it's as big a deal as this. To read English, you need to know to look at the words on page starting on the left and moving to the right, okay? That's the kind of big deal this is. Going 3' to 5' on DNA is going to mess you up as badly as reading from left to right if you're trying to make sense of English, okay? Same level of deal. That orientation, it's just as important as left and right. And just as the left side of the page is the beginning in English, 5' is the beginning for DNA. Everything else comes after the 5' end. That's where you start writing. That's where you start synthesis of DNA. Information is essentially needs to be interpreted from the 5' end to the 3' end. So there's DNA, double-stranded, anti-parallel strands, 5' to 3' in one direction, 5' to 3' in the other. There's information here. DNA is all about storing and passing on information. It's not about using that information. It's great for storing information, the redundancy. If you bump into something and get jostled a little bit, you can repair using the information on the other side. Cool. Watson and Crick passed this idea off that they figured if you separated the two strands, you could replicate the chromosomes, and that would come in handy for cell division and even organismal reproduction. But DNA isn't about using that information. RNA is what we turn to for the use of that information. And so you've got information stored in DNA, but you need to get it to something that can allow it to be put to use. You need to get it into RNA. And so how do you do that? We use an enzyme called RNA polymerase to do transcription to take the information from one of these two strands and use it to make an RNA copy of the other strand. Check it out. What we've got here is an RNA copy of this bottom strand. The RNA polymerase is using the information in the top strand to make an RNA copy of the information in the bottom strand. Everything is exactly the same in the RNA copy as it is in the bottom strand of the top here, bottom strand of the DNA here, with the exception, right, that T is replaced with U. A subtle difference between DNA and RNA. But otherwise, everything else is the same. If you have a T here, you got a U there. You got a G here, you got a G there. G here, G there. T there, T there, right? So it's a perfect copy of what's been going on in that bottom strand of the DNA molecule. And it, too, has a 5' end and has a 3' end. You know how the RNA got synthesized? It gets synthesized from 5' to 3'. The RNA polymerase starts here. That's the first nucleotide it brings in, and then it adds in all the rest, moving in that 3' direction. So now we've got information in RNA, and we need to be able to get that information as it's spelled out in RNA into proteins. Ribosomes are the thing that do that job of translation. Logistically, how's that got to happen? Logistically, when people first wrestled with this idea, how can there be a correspondence between the information in RNA and the information that's used to make proteins? When people first started to wrestle with this idea, the suggestion was made, the obvious first suggestion was, for every nucleotide, there must be an amino acid. There's a one-to-one correspondence, right? If you have a U, maybe that means you plug in a tryptophan. It doesn't take too long for somebody to raise their hand and say, hey, wait a minute, that can't work, because there are only 4 nucleotides and there are 20 amino acids. So there can't be a one-to-one correspondence between nucleotides and amino acids. That can't be what ribosomes are looking at. We know there's information in the sequence of nucleotides, but it can't be a one-to-one correspondence. How about a 2-to-1 correspondence? Maybe ribosomes look at 2 nucleotides at a time, right? How many different combinations of 2 nucleotides could you have? You could have, here, I'll get you started. You could have AA, you could have AU, you could have AG, you could have AC, 4, right? You could have UU, you could have UA, you could have UG, UC, that's 8. And I tell you what, we're halfway there, because there's only 2 other nucleotides that I can put first in the pair, right? There are 16 different ways that you can combine 2 different nucleotides, if you only have 4 nucleotides to work with. So a single nucleotide code can't work. It only gives you 4 single nucleotides, and you got 20 amino acids. A double nucleotide code can't work, because it only gives you 16 combinations. And I got to come up with something that lets ribosomes be able to recognize 20 different things. What are we going to do? Well, let's think about what might happen if you use 3 nucleotides. A triplet genetic code. Most of you probably already know that's what's really going on. Ribosomes are looking at 3 nucleotides at a time to determine which amino acid to add. There are 64 different ways that you can combine the 4 nucleotides. 64, finally, we've got a number that's bigger than 20. If you look at 3 nucleotides at a time, there's an opportunity for there to be a specific set of 3 that says tryptophan, another set of 3 that says alanine, another set of 3 that says glycine, phenylalanine, serine, and all the rest. So there's a triplet code that the ribosomes are looking at. Those triplet codes are called codons. So 3 nucleotides in a row that a ribosome looks at, that's a codon. Each codon spells out a different amino acid. It orders the ribosome to put in 1 amino acid as opposed to any of the 19 alternatives. And it's in that way that information stored in DNA gets used to spell out a specific sequence of amino acids for a protein. All right. So there's a triplet code. Doesn't take too long for people to figure this out. Once you got the structure of DNA, you're able to say, okay, it's the sequence of the nucleotides that must be involved here. Triplet code, 64 different codons. What do each of those codons tell ribosomes? Curious people want to know, right? What's the set of 3 nucleotides that a ribosome needs to see in order to understand, put a phenylalanine in now, right? What are those 3 letters? Brace yourself. Here is the answer to that question. This is the genetic code, okay? What you see there is there's a spot for each of the 64 different ways that you could combine the 3 different nucleotides. One of those 64 is UUU, right? Uracil, uracil, uracil. Turns out that answers my question. What's a ribosome need to see in order to get the idea to take the command, put a phenylalanine in here? UUU will get the job done. Another opportunity for a triplet, one of the other 64, is UUA. When a ribosome gets the signal UUA, when it sees that codon, it plugs in the amino acid leucine. When it sees AUU, it gets a different message. The message now is use the amino acid isoleucine. All right, so there's 64 of these 3-letter combinations. And the way this table is set up is pretty straightforward. Here you're looking at the first letter. Look at this. All of these 4 start with the letter G. All of these 4 start with the letter A. All of these 4 start with C. All of these 4 start with U. The next one here, U, the second one is U, 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 U. All of these have U for their second position, right? And the third one is just all the different combinations that you have, the 4 different combinations for each of those first 2 combinations. So that's the genetic code. At least a couple of you have to be wondering if you might be able to enjoy, over the course of the Thanksgiving break coming up in a week or so, enjoy the opportunity to memorize the genetic code. You guys up for that? You want to sit down on Thanksgiving, right after dinner, commit all that to memory? What a fun, you would remember that Thanksgiving for the rest of your life. Guaranteed, right? This is going to be perhaps your single most memorable Thanksgiving ever, and I don't want to deprive you of that joy, but I'm not going to make you have that joy. That's something you have to take on yourself, right? Because I'll tell you what, not many people in the business have that genetic code committed to memory. This is the kind of people who like to memorize pi to like 500 digits. Those are the people who memorize the genetic code. The rest of us, myself included, I need to know what a triplet codon spells out. You know what I do? There's an app for that, right? There's a website for that. There's a table in your book for that. The genetic code shows up all over the place. But it would be nice if you knew how to use one of those tables, right? So I can imagine putting on your final exam that table and giving you a string of nucleotides and asking you to, this is the fancy term, conceptually translate it, right? For this particular string of codons, what polypeptide would you make? And that would require you to be comfortable with using a table like this. So I think that's a good idea. For those of you who want to commit the whole thing to memory, have at it. More power to you. I'll cheer for you, and I'll be really impressed when you come by my office hours and I quiz you on it and you give me the right answer every time. That's just swell. Good for you, right? So, but there's the genetic code. There's stuff about the genetic code that you must have been noticing as I've been prattling on about memorizing the genetic code. For instance, how could you have missed the fact that there's more than one way for a ribosome to get the idea to put a phenylalanine into a growing polypeptide? Did you think I didn't notice that? That's a big deal too. There's at least 2 ways that ribosomes get the message phenylalanine. It turns out that the codon UUU and the codon UUC both say precisely the same thing to a ribosome. They both say, plug phenylalanine in here. All right? There's a word for that. We've got a redundant genetic code. For many amino acids, for most amino acids, there's more than one triplet codon that tells the ribosome, use this particular amino acid. Right? So you can see there are 2 for phenylalanine. There's actually 6 codons that are equivalent that tell ribosomes use the amino acid leucine. You must have noticed this one. I mean, it's color-coded to pop out and make you think, what's up with that one here? Well, AUG, the triplet codon, says use methionine, the amino acid methionine, to a ribosome. It also says this is a good place to start protein synthesis. So most proteins begin with a ribosome settling down on an AUG codon and bringing in a methionine. Most proteins begin with the amino acid methionine because in addition to coding for methionine, This is also referred to as a start codon. In the English equivalent of a start codon, it's the capital letter at the beginning of a sentence, right? The letters you're writing down on pages, the letters you see in a textbook, they're storing information. DNA is storing information. It's the sequence of characters that has that information content associated with it. The individual nucleotides are like individual letters. The codons are like words. The combination of codons is sort of like a sentence or a paragraph, right? It's a cohesive, useful chunk of information that can kind of stand on its own and do something kind of useful, right? So that analogy works really well. You could get by if your textbooks didn't have capital letters at the beginning of sentences. For some reason, the publisher wanted to save a couple pennies and not buy capital letters for their typesetting machines, right? You could get by, but it's nice to have them, right? It helps you find where the beginning of a thought is. It helps you find where the beginning of AUG, capital letter at the beginning of a protein, okay? Functionally more important, perhaps, than the capital letter at the beginning of a sentence is the period at the end of the sentence. That comes in handy, right? It's nice to know that's it. That's the end of this idea. Ribosomes need a specific signal to tell them you're done, stop translating, move on, and start to make a new protein. Well, there's a codon for that. There's actually 3 codons, cleverly known as stop codons. These 3 codons, UAA, UAG, and UGA, give an unambiguous signal to a ribosome. They tell it, stop translation here. There is no amino acid that corresponds to those 3 particular codons. Instead, what the ribosome gets is the information. It gets the idea, stop, terminate translation, finish making this protein, move on, and try to start working on making another protein. So again, more or less functional equivalent of the period or maybe an exclamation mark at the end of a sentence. But that's it. That's the genetic code. Something that I think is awfully cool is the fact that that's the genetic code. That's not just the human genetic code, right? That is the human genetic code. It's also the dog genetic code. How about that? And it's not just the human and dog genetic code. It's the wheat genetic code. It is the yeast genetic code. It is the bacterial genetic code, the billions, trillions of different kinds of bacteria. Every darn last thing on this planet uses one genetic code. That's it. Is that cool or what? That's one of the strongest arguments that I think I can personally point to in support of the idea that we all share a common ancestor. Early in the history of life, when things were getting sorted out, you could have had GGG make glutamic acid. Instead, by chance, GGG got used for glycine, and everything from that point on has followed that same rule. It's a universal genetic code, and that's the fancy word that people use to describe this. It's not just the genetic code, it is the universal genetic code. There are 20 different amino acids. All of them get spelled out with the same set of rules. Now, I'll tell you, that's the freshman biology story. There are some organisms, obscure, relatively uncommon, that use more than those 20 amino acids. It's a fun research project. If you want to do an independent study someday, do some research on those. There's like another 3 or 4 amino acids that some living things use beyond the normal set of 20, and they've carved out some of the genetic code for those, right? So there are a couple of relatively minor but interesting exceptions. For all intents and purposes, though, unless you're really getting hardcore into molecular evolution, this is the universal genetic code. So there's some built-in redundancy. There are more than one way to say, I want to use phenylalanine. There's 6 ways to use leucine. None of them have more than 6. There's 2 that have 6, right? It's leucine, and what's the other one? It's around here somewhere. See? Those of you who are going to memorize the genetic code can tell me. Maybe there are only one. That's, nonetheless. But so you can see 6 here for leucine, 2 for phenylalanine, 4 for serine, 4 for proline, 1 for methionine, 3 different stop codons. Here's tryptophan. Is it all by itself? Yeah, look at that. Tryptophan has just 1 code, so like methionine, tryptophan has just 1. But you got the whole shebang. There's something I'm just dying to tell you. I'm not sure how important it is for you guys to know at this point, but it turns out, remember, there are, what, 4 different kinds of amino acids? There are the hydrophobic amino acids, the hydrophilic amino acids, the positively charged amino acids, the negatively charged amino acids, right? Remember, there are 4 different kinds. If you were designing a genetic code, right, if I gave you that as a homework assignment, I think you should get extra credit if you come up with the idea of having similar codons spell out similar amino acids, right? Because that sort of builds in a nice little safety feature for you. If you have a mutation at the level of DNA that changes a nucleotide, wouldn't it be nice if the new amino acid that the codon was coding for was similar to the original amino acid that's less likely to mess you up, right? Guess what? That's how our genetic code got put together. God gets some extra credit points for this particular genetic code. If you want, take the time. There's wonder to behold as you dive into the genetic code. Similar amino acids like leucine and isoleucine, they're about as similar as you get, right, have similar triplet codons. And that builds in sort of like an error protection as well. If you have a mutation, it may not be that horrible for you if you substitute a leucine for an isoleucine. And you can see, again, they're kind of clustered together. So I brought up this nasty word of mutation, errors. Let's talk about specifically how those errors can play out. One of the first errors to really get characterized thoroughly and carefully is the error that's associated with sickle cell anemia. For those of you who don't know, can there be somebody who doesn't know? Sickle cell anemia is a genetic disorder. It gets its name from the fact that red blood cells, everybody knows sort of the iconic image of a red blood cell, an erythrocyte. It's like a little red donut, kind of dimpled in the middle and tubular around the edges. It's like a little red donut. People with sickle cell anemia don't make little donut-shaped red blood cells. They make red blood cells that have odd things pointing out of them. They look like the sort of sickle you might use to harvest wheat in the old days, right? They're kind of hook-shaped. Those hooks get caught up on each other in your bloodstream if you have this disease. And if they get caught up on each other, you can throw a clot. And those little blockages in your bloodstream cause you problems. At the very least, cramping and discomfort, potentially things much more serious like gangrene and amputation and all that stuff. It's nasty. It's not a nice disease. And so people were interested at the genetic level, what's going on. It turns out that people with sickle cell anemia have just 1 amino acid different in their hemoglobin proteins relative to the people who do not have sickle cell anemia. That 1 amino acid difference is due to a single nucleotide difference in one of the genes that codes for hemoglobin. So here, check it out. Here's that wild type phrase again. This is what most people have. If you were just take a random sampling of people, you'd find at the level of their DNA on one of the genes that codes for hemoglobin, at some point, there's a triplet codon that is GAA. Now, if you want to be, testing yourself, you can go to the previous slide. We know that wild-type people have GAA for one of their codons, right? What amino acid does that code for? Well, the first one was G. The second letter is A. Wait, there it is. The second letter is, so we got G, A, A. All right? So first letter was G, second letter's A, G, A, and the third letter happens to be A. That codes for an amino acid. 3-letter abbreviation is GLU. That's glutamic acid. This is one of the negatively charged amino acids. Cool? Okay. So we thought GAA might code for glutamic acid. Pretty good, huh? It's like magic. This stuff all sort of hangs together. So in normal hemoglobin, at this point in the polypeptide chain, you've got this negatively charged amino acid that we call glutamic acid. In people with sickle cell anemia, there's a single nucleotide different at the level of their DNA. That first A, instead of being an A, is a T. That's it. In every other respect, they're exactly the same as everybody else. But if you got sickle cell anemia, instead of having that second letter be an A, it's a T. That means at the level of RNA, it's a U. So that codon now is GUA. Okay. GUA. Let's go back to the other slide. GUA. That tells ribosomes to use a different amino acid. Specifically, it's saying use valine. You might recall valine, probably not, but I can entertain the idea. You might recall valine is a pretty different amino acid from glutamic acid. Valine is one of those hydrophobic guys. Glutamic acid is negatively charged. You're putting in a fairly different amino acid, and as a result of that, the protein behaves differently, and you end up with sickle cell anemia. I'll tell you specifically what's going on. This glutamic acid on the hemoglobin protein, it's on the surface, as you might expect, right? It's a negatively charged thing. It's going to be water soluble, all that stuff. It's on the surface. One hemoglobin protein doesn't like to rub up against another hemoglobin protein at that spot because those two spots both have a glutamic acid and the similar charges repel each other. If you substitute glutamic acid, though, with valine, it's still on the surface of the protein, but now you've got a hydrophobic pocket on the surface of one hemoglobin protein that likes to butt up against another hydrophobic pocket on another hemoglobin protein, and you make these long chains of hemoglobin proteins that make these strange protuberances out of the surface of the red blood cell that give you these hooks that cause the whole phenotype and problem associated with the disease. All right? What we've just talked about here is a mutation. All right? We've got the wild-type hemoglobin. We've got a mutant hemoglobin. And this is actually a special kind of mutation in the sense that it has caused the amino acid that's coded for to change. All right? Because you could have had a change. Let's check out this one. What if we had changed, instead of the second letter, instead of the second position in the triplet codon, instead of changing that A to a T, let's see what happens if we change this A to a T. Okay? So not this one A to T, this one A to T. So now I've got GAU. All right, let's go back over here. GAU, right? It's a substitution of the amino acid. Instead of using glutamic acid, I'm using aspartic acid. But you know what? Functionally, they're very similar. These are two negatively charged amino acids. You're not going to have that hydrophobic pocket going on. So that is a substitution at the protein level. But it's not as big a deal kind of substitution as what you have over here. What if we had changed the A to a G? So now instead of GAA, it's GAG. GAG. Whoop. GAG and GAA are the same. So that's not even a substitution. That's a substitution at the nucleotide level, but it is not a substitution at the amino acid level because the amino acid is staying the same. What kind of phenotypic problems would somebody with GAG for that codon have compared to somebody with GAA? Kind of trick question, none. We wouldn't expect, at least, there to be any, right? Because at the protein level, the proteins got a glutamic acid. You got a glutamic acid. What's not to like? So notice how some changes in the nucleotide sequence don't even result in a change in the amino acid sequence. And again, that's because of the redundancy of the genetic code. All right, so you've already got a sort of subliminal peek at the next slide. The part of the beauty of the genetic code, being a universal genetic code, is you can do cool stuff like this. All right? You get me a gene that codes for a protein that glows in the dark, right? And you know there are such proteins, right? You've all seen fireflies, right? You may have seen jellyfish that fluoresce at night, right? You get me a gene that causes a protein, that makes a protein that glows in the dark, and I can take that gene and I can plug it into any old organism that I like, and if I can get that organism to make a protein off of that gene, I don't have to change the nucleotide sequences of the gene at all. It's going to make the same string of amino acids, the same polypeptide, in a human or a pig or a tobacco plant as it did in the jellyfish or the firefly. How about that? And I don't know if you can tell. I'm going to turn the lights off so you can see what's going on here, but I want to make sure you appreciate. That pig is glowing, guys. All right? That's seriously a glowing pig. You can't do that if you don't have a universal genetic code. Little bit of knowledge, crazy power, right? That pig is going to glow for the rest of its life, whether or not it eats that glowing plant. And by the way, speaking of glowing plants, you must have seen in pet stores the glowing fish, the zebrafish, right? If you've seen the fish, did you stop to look at how much they charge for those fish? It's like $5, $6 for a glowing zebrafish. The regular zebrafish are like 25 cents. I'm starting to sound like Bernie Sanders here. I don't know. But what's up with the extra? If you can charge 10 times the cost for a glowing zebrafish, you guys can retire in a couple weeks if you could figure out how to get the glowing gene out of the tobacco plant over there and have it be in like a rose bush, right? Think about the sales at Valentine's Day, for glow-in-the-dark moon gardens take on a whole nother meeting. There's no reason you guys can't do that. In fact, that might be a fun undergraduate research project right there. All right, so that's my gift to you. I gave you the joy of memorizing the genetic code over Thanksgiving, a memorable Thanksgiving. Now I'm taking care of your financial well-being. All you got to do is get some of these genes expressed in something more interesting than zebrafish. And for goodness sakes, if you can get it expressed in a pig and you can get it expressed in a zebrafish, roses are super easy, right? That shouldn't be a problem. So, more about the genetic code. Well, better yet, more about the whole process of the central dogma. Let's do this. Let's take a closer look now at what's actually going on with transcription. Okay? We'll do this one step at a time. What's happening with transcription? I've told you in sort of broad terms what's happening with transcription. And I told you that this 5' to 3' thing's a big deal. Let's talk about that. So here's what's going on. You got a double-stranded DNA molecule, 2 parallel lines, DNA. Let's say that from here to here is the coding sequence that you need to spell out a protein to make a polypeptide. We can string together certain amino acids to make a protein, starting here, moving to there. But we got to make an RNA copy of that gene to do that. So you start at one spot on the DNA sequence that gets a fancy name. We call that spot where transcription begins, the promoter. Okay? So this is where transcription is going to start. Remember, the enzyme that does transcription we call an RNA polymerase, so it needs to sit down and recognize a promoter. We'll talk about how it is that it does that in a week. But the RNA polymerase sits down on a promoter, and when it does, it needs to start sniffing out the information stored in this DNA molecule, right? That information stored up in the DNA molecule is kind of tied up. it's involved with those hydrogen bonds between one strand and the next strand. What's there for the RNA polymerase to sniff? Nothing. It needs to separate those two strands so that it can get a better look at the nucleotide sequence and that information stored in the nucleotide sequence. So the first thing that has to happen when you want to do transcription is an RNA polymerase needs to come along and find the start of a gene. We're going to call that a promoter. Once it finds the promoter, it needs to then start to look in detail at the information stored in the DNA. And to do that, it needs to separate out the two strands. It needs to, and here's the fancy word, it needs to denature the DNA. So it separates the two strands. And then it needs to begin making an RNA copy of one of those two strands, right? So it separates the two strands, and it begins to make an RNA copy. Check out what it's doing. The RNA polymerase, as it's moving along, is really just using information on one of those two strands. It's not paying any attention in any way to the information on the top strand here. It's using just the information on the bottom strand to bring in new nucleotides to make an RNA copy of that double-stranded DNA molecule. Okay? And notice further the direction in which the RNA polymerase is working. I'll cut right to the chase for you. I'll tell you the bottom line is this. It's all about the baby RNA molecule. It's all about the new RNA molecule. It's all about the world seen from the perspective of the RNA molecule. So notice the new RNA molecule, it starts to be made at its 5' end, just like we've talked about earlier today. And you extend that molecule off in the 3' direction. If that's the case, remember this new RNA molecule is going to be complementary to one of the two strands of the DNA molecule. It's going to be anti-parallel to one of those two strands. It's the RNA, what that means is the RNA polymerase is using the information on the bottom strand going 3' to 5' from the perspective of that strand as it makes its new copy of the RNA molecule. All right? So orientation here is important. Unless somebody goes way out of their way to tell you otherwise, RNA synthesis is 5' to 3', always. You can't even go out of your way to say other than that. RNA synthesis always starts at the 5' end and moves to the 3' end. And when people are talking about the direction the RNA polymerase is moving, they'll always be talking about 5' to 3', unless they go way out of their way to tell you they're talking about it from the perspective of something other than the RNA molecule. So here I could say the RNA polymerase is moving 3' to 5' relative to that bottom strand of the DNA molecule, but that's just going to mess you up, right? And so rather than say that, I'll say the RNA polymerase is moving 5' to 3'. And you fill in all the rest. It's moving 5' to 3' relative to the top strand. It's moving 5' to 3' relative to the RNA molecule, which is really where the focus is. It happens to also be moving 3' to 5' relative to the template strand. The strand that the RNA polymerase is using is the template for that new RNA molecule. So the RNA polymerase chugs along, 5' to 3'. Eventually, it falls off. The DNA goes back to being double-stranded, and you've got yourself a new, shiny RNA molecule with a beginning at its 5' end and an end at its 3' end. Conceivably, this will contain within it a bunch of codons that can then be used by ribosomes to make a protein. Here's a zoomed-in version of what's going on inside of the RNA polymerase. Again, notice you're separating out, you're separating those two strands so that the RNA polymerase can bring nucleotides in to see if they base pair well, if they make the right kind of hydrogen bonds. Again, the direction of transcription is 5' to 3'. I think perhaps the most interesting thing. It's subtle. I don't know that it's a particularly important point, but it's nice to sort of have an idea of what's really taking place here. Perhaps the most interesting thing about this particular picture is just the scale of things. The RNA polymerase is denaturing not the whole gene, right? And not just one or two nucleotides. It's actually separating out on the order of 10, 15 nucleotides. That's all that's being denatured. And all of those nucleotides are inside of the RNA polymerase as the separation is taking place. Right? So it's a small thing, kind of a little bit of trivia for you there, but I'd like it if you had a sense of how big a region was being denatured. That could come in handy for you in other contexts. And again, this is just a zoomed-in version of what we've been talking about for the last 5-10 minutes. What we've just talked about, what you've just seen, that's what's going on in prokaryotes. It's also what's happening in eukaryotes. But a while ago I told you transcription and translation happen simultaneously in prokaryotes. Transcription happens in the nucleus in eukaryotes. Translation happens in the cytoplasm in eukaryotes. And I tried to make a big deal of the fact that we can process our information content. As eukaryotes, we can process the information at the level of our RNA molecules such that it can be different than what's spelled out within the genes at the DNA level. There are three kinds of processing that eukaryotes do to their RNA molecules. And this slide is giving you some names for each of them. We change the beginning of the molecule. We put a 5' cap on. So instead of having that first nucleotide be the nucleotide the RNA polymerase brings in, we'll tack on an additional nucleotide. That gets called a 5' cap. Also, another kind of eukaryotic processing is we change in a big way the 3' end of the RNA molecule, cutting off a big chunk at the end and replacing it with a bunch of A's on the order of 200 A's. It's called a poly-A tail. Good name, right? A couple hundred A's get tacked on to the 3' end of a eukaryotic RNA in place of whatever it is was encoded at the level of DNA. But the biggest change by far of all, the one with the greatest consequence by far, is this idea of splicing. And what happens with splicing is eukaryotic RNA polymerases sit down on a promoter, they transcribe through a gene, but along the way they transcribe things, these little salmon-colored boxes, they transcribe things that don't end up in the final messenger RNA that ribosomes use to make a protein. They transcribe two different kinds of things. They transcribe the exons that end up in the final messenger RNA that ribosomes use to make proteins with. And they also transcribe introns. And the introns get cut out within the nucleus, tossed away within the nucleus such that ribosomes never see them, and the protein that gets translated comes just from the exons and not from the introns. That whole business of editing the internal content of RNA molecules, again, exclusively a eukaryotic thing, that whole business is called splicing. Okay? I think that's plenty for today. So what we'll do on Thursday is talk a little bit more about mutations. I'll show you some more about how splicing plays out and the implications of splicing. But it's going to be fun, and I'll look forward to telling you more on Thursday.